Welcome to ISL-enabled Virtual Lab of CSIR.
Hello students, I am Dr. Archana Singh. I am a scientist here and I work on a pigmentation disorder called vitiligo, wherein some people lose their pigmentation on the skin. This is a normal skin of a vitiligo patient, and here the melanocytes, the melanin content is actually normal in the normal appearing skin. However, if you look at their vitiligo skin, you absolutely see zero melanin content in there. So this is what we are trying to understand. Once the melanocytes are lost from these patients' body, what really happens to the other cells. This is called a compound microscope, which is nothing but a kind of a light microscope. So the source of illumination there is light, and you usually, I'm sure you must have visualized samples like root tip or cheek cells on that, and you must have seen the cell boundaries very much clear. You can also see the nucleus, and I'm sure you must have seen these kind of samples in your laboratory. You see this image. What you see here is a typical Golgi apparatus. You can see the cisternae stacked, and these small things are the mitochondria. Let me just show you some more pictures. This one. So this is a typical mitochondria that you can see. So you have the outer membrane, you have the inner membrane, and now these are the cristae of the mitochondria. In a nutshell, if you want to know, is that it magnifies a given object while still retaining the clarity. For example, if you look at any of your images that you take in the camera of your phone, so when you try to enlarge it or magnify it, what you see is that the picture starts to get pixelated or you lose the clarity. So here it magnifies to a really large extent, but it still does not give you that pixelation or the blurred images. So the clarity is absolutely maintained. The typical magnification, first I'll talk about the magnification. The typical magnification that you see under a light microscope or a compound microscope as you call it is 10x, 20x and 40x objective lenses. Most of the microscopes provide you these three objectives. So, and there are some microscopes which can go even up to 100x using oil. This one actually gives you up to 7 lakh x magnification, 700,000 times the original size. The x here is only 5,000. So even at a very small magnification, in terms of time I am talking, it's a small magnification, you can see that even at 5,000x, you are able to see each and every cristae of the mitochondria beautifully. So you don't really need 7 lakh x for everything. For very small objects, like for example viruses, we may need to go up to 7 lakh x. But for most of the routine cell biology stuff that we see, the cells, the mitochondria, the Golgi apparatus, they all can be visualized at less than 5,000 x. But it is not just the magnification. As I told you, you can just keep on going magnifying your images, but that is of no use till you are still able to retain the clarity or the resolution. Okay, here the resolution here is actually 0.2 nanometer. That means the same two objects, even if they are only 0.2 nanometer apart, this system can still tell you that yes, they are actually 2 points and not 1. So in terms of resolution or clarity, the compound microscope gives you 200 nanometer. Here it gives 0.2 nanometer. These different circles, these are 3 millimeter circles. This is equivalent of a slide. This grid on which we keep the sample now goes on this grid holder. Okay, and if you see it very carefully, you will see a very small tiny hole which is exactly 3 millimeter, which fits this grid onto this, and once you place your grid, which now contains your sample, this goes straight into this place, and this goes all the way down into this, and what you can actually see after inserting it is just this. The entire rod goes inside. So basically it goes somewhere in the middle of this, and this is the long tube that you see is the path of the electrons. So this is a long column that you see, and at the top of it, just like in compound microscope you have light as the source of illumination, here the electrons are the source of illumination and they are at the very top. So there is a source which emits electrons. The column is very special in the sense it has no air. It is a high vacuum column because the energy, otherwise the energy of the electrons will get dissipated. Electrons travel best in vacuum. Okay, so this is a long column because it needs a long time or a travel time to get accelerated. And in this system we can have tungsten as the source. So as soon as it passes the sample, the electrons are transmitted through the sample. That means it enters and then comes down. And therefore the name transmission electron microscope. So the electrons are moving and finally bombard your samples, and the images formed here. What you see here is a fluorescent screen, but these days we really don't use the screen per se. We directly transfer the images onto the digital media. This is a skin sample, a human skin sample, and what you see is two cells here. Now how do you know these are two cells? Because you can see one nucleus here, this in a grey line, and the other one is here. So these are two cells, and what you see in between, please don't confuse this as the nucleus. This is the nucleolus. Okay, so nucleolus followed by nucleus, and then what you see here is actually the cytoplasm. And skin, as most of you would be aware of, contains a lot of pigment, the melanin pigment, which you see as this black pigmented electron dense granules. So this is what gives our skin the characteristic color.